The point is that the water, even in these simple cells, is trapped and will form local areas of symmetry which can be broken. Now the next one will show you what happens with the DNA. The DNA, if you look at it from this side, it is not a nanotube. But if you look at it from above, please, the next one, then you see there is something like a tube. Water will go inside here because it is all swimming in water. And water will go inside here and you have an effect like in a nanotube. Whereas when the DNA is coiled up tightly, this is the left case, the water cannot go inside. And actually this is a site where the DNA is red. There's a reading. And if there's a reading, the DNA has to open. And when it opens, the water will go in and can form regular patterns of so-called nano-ice. Yes, next one is, yes, now the microtubuli come. Microtubuli are formed in cells on all different levels of living cells. For example, you have many cells with a flagellum. Please. Chiranjeev? Okay. There are many cells, even bacteria, which have a flagellum, which propels them, which allows them to move. And each flagellum is made of microtubuli. This would be the uppercase. Then each cell, each eukaryotic cell has to divide. Now you will have a set of chromosomes. The chromosomes will double. And then that doubled set of chromosomes has to be pulled to the two sides. Then it will divide. And that pulling of the chromosomes to the one and the other side, it is done by nothing but microtubuli. The microtubuli are produced in short length. Then they will polymerize and form a thread. The thread will fix at a particular site, which is called the centromere on the chromosome, and it will fix on the centriole, which is the pole body. So there's a centriole. It is very interesting. The centriole is made of two short microtubuli. One microtubuli like this, one like this. This is a centriole. And the microtubulus will be fixed at the centromere. It will go here, it will fix at the centromere, one particular site on the chromosome, and it will fix on the centriole. And when this is done completely, all the chromosomes are fixed to the centrioles, then it will pull the chromosomes to the poles, and then the membrane can come in and divide the two cells. And that process is highly important. If it fails, the cell will probably die. If one cell gets one or two chromosomes additionally and the other cell lacks one or two chromosomes, then this cell will not be able to survive. So that process has to work, and it is a very basic process for the life. There is no division of cells without that process. Okay, that was just a spindle. The spindles in the cell division are made of microtubes, microtubuli. And finally, the same stuff which is found in bacteria to form a flagellum, which is found in every eukaryotic cell to enable a cell division. That same material is used in the nerve cells. Now this nerve cell, it has a cell body, and then it has an axon, and it has the dendrites. The dendrites are like the branches of a tree connecting to the neighboring trees, and the dendrite is like a deep, deep root. Now, all these dendrites and the axon, they have to have some stability. If they would be just protoplasm, that is too soft. They would have no stability at all. To stabilize it, they are fully packed with microtubuli, with long microtubuli. So this would be a dendrite, it would have more dendrites, and this would be the axon, and it is fully packed with the microtubuli. Now these microtubuli, next one please, this is another picture how these microtubuli are stabilizing the whole cell. And the microtubuli are interconnected among each other by protein bridges. So that means that the space for the water is not narrow only within the microtubulus, it is also narrow around the microtubulus. So the space is, the water is organized or in a symmetric array inside and outside of the microtubuli in an axon or a dendrite of a nerve cell. I mean, our new point in that respect is that we are replacing spontaneous symmetry breaking by organized symmetry breaking. But the rest has been published already something like 8 or even more years ago. Jibo and Jazue from Stanford University, they published these things in the late 90s. And it is recognized by the Stanford Encyclopedia to be the best model of how the brain works. And when we think about how the brain works, we have to realize that you can operate a big part of the brain and it still works. So if the various parts of the brain are very essential for its operation, then this should not be. Rather, it can be replaced. So we have, yeah, I mean, we know patients that they got a bigger portion of the front lobe, or almost whole front lobe to be removed, and still they remember the things and live. So the brain is so flexible, and it seems to have some holographic features. So what is present here is also present there. And if you remove a piece, it is available at another site. That has some limitations, of course. I mean, if you remove too much, anyhow. So now we come to the physics of that, I hope. Oh, no, yet one more picture. Yes, the next one. Yes, no, previous. That one. In between. No, you are going backward. Next one. This one. This one again shows in an electron microscopic high resolution. This would be a microtubulus. Here it is dissected, so you see the inner face of a microtubulus.
How big is the bar? How big is the bar on the left bottom left?
Huh?
How big is the scale bar on the bottom left?
The scale,
It cannot be a dissection of one microtubule. There's no TEM is not so. There's no microscope. Will give you that. That cannot be.
Why?
Why?
No,
You're familiar with the instrumentation.
No, scanning a cell will give you down to atomic scale, so it'll easily do this. But what is the scale? There's no scale.
it is not written. It will be in the text below, and that's not presently available. But there is the internet address given here, and we can just reconfirm what is the scale.
We looked up there, didn't we? It was about 20 or 30 microns.
We went after this. Yes, something like that.
Yes.
But what it shows that the spaces around the microtubuli are also quite narrow by these proteins linking them. So that is my point, that within the tube, the water is in a nano-ice formation, that's clear. But around the nanotubes, the spaces are also very tight, very narrow, and the water cannot do what it wants. Rather, it will be in a fixed position, quite fixed position.
So this is a picture of, remind us what this is a picture of?
This is an electron microscope picture of an axon. Means lots of microtubuli are running completely parallel to each other, being interlinked by these proteins, by these proteins here.
So this is right inside one of the strands.
Yes.
An axon strand inside the brain. So you cut it open, looking inside it.
If you go one picture back, you see it in a schematic way. This would be such a cut. Okay?
If you think of nano ice, you don't have to think of 0 degrees or below, but 35 degrees or 37 degrees.
Yes. Once you start constraining something, the physics of that thing changes. So as you make something small, it becomes even a solid, even at
Yes.
temperature.
Yes.
Yes.
Yes, it becomes practically a solid.
Solid.
Yes. Because you concept the physics of a small thing is different than a big thing. So you have surface effects which modify the solid liquid.
Now here are our numbers. Yes, this is a model which was created, and here you get the numbers. This is the model of the microtubulus, and the numbers are taken from cells. So it means what it means. The diameter, the inner diameter of that tube would be 22 nanometers. And the thickness of it will be 4 nanometers. No, the inner diameter, sorry, it is 14 nanometers. 14 nanometers. The outer diameter would be 22 nanometers.
And water's what, a few angstroms, about a nanometer, isn't it, the more the size? Is that right?
Approximately 1, yeah.
It's about 14, which is pretty weak, quite small. Smallest things we can make, 3 nanometers, and that's...
But the inner surface is mostly made of carbon atoms, mostly. Of course, hydrogen, oxygen, that's also mixed in, but the matrix is carbon. And probably it will keep the water in some distance because of some electric effects.
Yeah, so you're looking, but you see at that sort of level, you're talking about 10 water molecules across. That's pretty small. So
Yeah.
you're talking about something. And then when you start thinking about the ring, which you showed before, the diameter of that is going to be, that's about, what's it going to be? It's going to be about 5, isn't it? So you can maybe get 2 or 3 of those tubes running down there. That's
Yes,
it, or 4.
yes, yes,
It's nice symmetric.
yes,
4,
yes.
4, isn't it?
Yeah, yeah.
That's exactly what it's going to be. So there'll be 4 tubes of water going down through that in that beautiful shape that Manohar showed before. So the water molecules are they're all polar. So you have polar, 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 so you get a ring. And that ring goes down.
Yes.
And there's roughly room for 4 of those.
Hmm.
I think if we're guessing the sizes right, for these molecules, that leads to about 4. But anyway, a small number.
Yeah, yeah. Now, I think now we have got an animated graph. That's one possibility how it might go. There has been sufficient work on that. It's very interesting, and they depict many possibilities how it can happen. I mean, such a wave front would run across that symmetry. Symmetry is broken, again it comes back to the original position, and the wave front runs across that line. And another, even more striking possibility would be the next one. It implies a rotation. Just see.
Oh, that's beautiful. Is that a jar graph?
Yes. That's very beautiful, isn't it? This I found something like 2 or 3 weeks after our last year's seminar. Because after the seminar, I felt something is missing. I must find something. And I got that. And that's the first time that I've shown it to an audience. So I'm very happy that I can show you that, because it gives an impression what actually happens. I don't say that it is like that. It all has to be investigated. It's only an idea how it might be. It might be something like that. It is not simply a front like we have just mentally seen it in the canal, but it might involve a rotation.
You see, the physics, this is beautiful, but the physics of it is wrong.